Now to chair the next session on future goals, I would like to invite Dr. Aditi Singh. She is a lab director at Advanced Center for Medical Genetics at Mahatma Gandhi Hospital, Jaipur, and Dr. Usha Gupta ma'am, professor and head Department of Gastroenterology at PGI.
Good morning to esteemed guests, seniors and friends. It is my distinct honor and also with deep admiration that I introduce a beacon of hope and compassion, Miss Stacie Lindsey. She's a creator of a non-profit organization known as Cholangiocarcinoma Foundation at Salt Lake City, Utah, the United States of America. Miss Lindsey holds a Bachelor's of Art degree in psychology from Brigham Young University, Utah, the US. As the saying goes, adversity introduces a man to himself. Miss Lindsey's profound journey began when her own brother faced the challenges of cholangiocarcinoma in 2005. She transformed her personal pain into a purpose by establishing a platform in 2005 that is committed to finding a cure and providing the quality of life for those affected by cholangiocarcinoma. Ms. Lindsey, you are an inspiration, really. So please join me in welcoming Ms. Stacie Lindsey as the speaker of today's session on advocacy for gallbladder cancer.
Actually, it is with an intention that I have included this, and I met Ms. Stacie Lindsey very recently at the Cholangiocarcinoma Foundation meeting in Mumbai, and Dr. Milind Javle introduced me to her, and then we were discussing that advocacy, especially patient participation in India, is very, very poor. We always deal with them as patients in the clinic, in the hospital, but once they are at home, then the other aspects of the disease and suffering we don't look after, and also the public participation. So with that intent, I discussed with her whether Cholangiocarcinoma Foundation would like to have a chapter on gallbladder also, but finally it evolved after discussion with few other people that it would be better to have a separate foundation. So recently during my visit to Chile, I discussed with the colleagues there, and I've had some very preliminary discussion with the Japanese and the Korean colleagues, and everybody is in favor of kind of joining such a kind of venture. So probably we will make a beginning soon. We will invite anyone and everyone who's interested in gallbladder cancer to join this. That is as far as the initiation is concerned, but any foundation then needs many other things, including logistics, financial support and all. But we'll see how it goes. So I thought it would be good to listen from her what Cholangiocarcinoma Foundation is, what it is doing, so that we can then adopt it to gallbladder cancer. So since again it is midnight in New York, she said it would be difficult for her to participate, but she's kind enough to send the video.
Foundation. I want to thank the organizers for inviting me today. I'm going to speak with you about advocacy for gallbladder cancer. I was asked to give you a little bit of the history of advocacy and how it works, and this will be particular to the US, but I hope there are some things that I talk about today that will inform you and help you think about how we can help you put together a gallbladder cancer advocacy group. So there are 4 main reasons to participate in advocacy. The first one, one primary goal would be patient support, education, and advocacy. Another would be political or public policy, so working with regulators, working with, for us, it would be the National Cancer Institute, organizations like that. Another would be research advocacy, working with the research community to provide patient perspective to the research process. And then another one is fundraising advocacy. Now, not all advocacy groups work on all of these different ways to advocate. And so one of the things that you want to be thinking about in your mind is, what is the first thing that you want to do? Where do you want to focus? So let me just talk with you a little bit about what an advocate is. So an advocate is usually a patient or a caregiver. A patient usually, and caregivers, when they are in treatment, usually do not know very much about their disease. Their main concern is their personal treatment pathway. What's going to happen next? Side effects, or clinical trials, those are the kinds of things that they're focused on, their own treatment pathway. Their focus is on themselves and their family, which is completely appropriate. And their emotions can play a significant role in their decision making. When you then look at what an advocate is, they are well educated and have more in-depth knowledge about the disease. Their main concern is influencing others through research or healthcare policy or through advocacy. They're focused on the wider disease community, not their personal experience, but the experience of the entire group. And their emotions are channeled into community objectives. So some patients will kind of cross this bridge over and become an advocate. And some patients will never cross that bridge. And that's completely okay. But there are ways for you to identify that, and we'll talk about that in just a minute. So what are the goals of advocacy? Advocacy is not about being politically correct or making patients feel good or included or checking a box. Advocacy has really tangible things, goals. Translating research faster and more effectively, getting answers more quickly, communicating progress clearly to the community, and involving the patient perspective in the process. So if we're all looking for better patient outcomes, then we have to start with better patient engagement. So advocacy organizations, mature advocacy organizations, can have strong research and technical expertise. They are primary educators and trusted communicators. They fund research and all kinds of research initiatives and efforts and projects. They have the ability to empower and activate the stakeholders or the constituencies that they're working with. And they have extensive engagement with patients, providers, regulators, and industry. So one of the important things as you start thinking about putting together a gallbladder advocacy organization is, where are the other aligned advocacy groups? So I wanted to share with you the Cholangiocarcinoma Foundation. These are some of the foundations that we work with. When we started, many of these groups were not in existence and have come about because many of them we have helped. We have given them access to information and to our specialists and to all the materials, to our website, and we're happy to do that. But this is just a look at some of those groups, and you can see they're from all over the world. So what does the foundation do? The foundation, the Cholangiocarcinoma Foundation, we are an organization that is 17 years old. And we have two pillars that we work from. One is patients and caregivers. We're connecting with the patients early and often. We're providing education and support. We facilitate patient and caregiver engagement in the community, and we're empowering the patients to participate in and advance research. The other pillar is research and breakthroughs. And so we're facilitating the availability of the global basic and translational resources. We're catalyzing innovation and progress. We're getting funds to high-risk, high-reward research and supporting long-term careers in cholangiocarcinoma. And we're trying to reduce barriers for companies who are developing breakthrough drugs and devices by working with the FDA. So let me just tell you a little bit about our newly diagnosed program. This is a program that has been in existence a little over a year. We have about 1,900 unique visitors into that portal each month. We have about 25 per week who sign up for a free care kit, and that care kit includes things that have been donated and resources to help them along their treatment pathway. To date, for the whole program, for about a year and a half, we have distributed about 2,000, a little more than 2,000 care kits. We have a specialist map on the website that helps them identify within their state who are the specialists, who are the surgeons that see a lot of cholangiocarcinoma and are really familiar with these complex surgical procedures, who are the clinicians, who are the rad oncs. So when they come to us and maybe they're in a state that does not have a lot of specialists, we can help them get to someone who can help them. We also have a lot of educational information, and we're working on getting it in multiple languages. We have a Cholangio Connect, a mentorship program. The mentorship program connects patients and caregivers with a mentor. And if you can imagine a patient, many of you are seeing patients regularly. A patient comes to you, they're very emotional, they can hardly talk. And they can't even probably hear what you're trying to tell them about their treatment options. The Cholangio Connect program is so that they have someone that they can talk through all the emotions. Maybe they have concerns about their family, their children, their parents, their work, all of those kinds of things that when you're seeing multiple patients per day, you may not have time to speak with them, but the mentors can talk with them. They've already been through this, so they can talk with them. We have made over 1,100 matches since this program started in 2017. We have a lot of clinical trial information on the website. We have virtual support groups. We have a Spanish-speaking support group. We have a bereaved support group. We have a patient support group, a caregiver support group. We have a veteran support group, and those run regularly throughout the month. We have educational webinars twice a month. We have a free nutrition guide for the patients, and also a free question and answer book about cholangiocarcinoma. It also covers gallbladder cancer as well. So those are some of the things that we are doing now, 17 years later. But when my brother was diagnosed, he was 38 in 2005. The end of 2005 was when he was diagnosed. And we started the foundation in 2006. And this is the landscape that we were looking at. No standard of care, no approved drugs, no clinical trials, no advocacy, no research funding, no collaboration of any kind that we could identify. And there were a few publications and not even any information on the website. So very limited public information that we could access to try and help him. So what does that mean? That means you've got isolated patients, you have limited or no resources, global resources, you have clinical research silos. So each institution just doing their own thing and randomly deciding what they think is important, limited research funding, and no progress. So here is where we are currently. We have an extensive network of patients and caregivers who are working together. We have a global resource mapping. We know and are working with resources and institutions all over the world. We have institutional collaborations. We have increased research funding, and we're making progress. And we know we're making progress because we've seen 7 FDA approvals for these patients. Almost $3.5 million in research funding has come from the foundation into research, and we'll do about 10 more research fellowships will be presented, about $500,000 in addition to this in just a few months at our annual conference. So gallbladder cancer is right here where we were when we started in 2006. And what we want to do is talk about a little bit about how can we solve this problem. We have a lot of gallbladder patients that come to our website, but we don't specialize in gallbladder, even though the specialists might be the same. We don't have specific information to gallbladder cancer, and we would love to be able to refer these patients to a group that could really help them. So how do you start? When you want to start a foundation, what do you do? So I'm just going to tell you what we did. And we started as a discussion board, which in 2006, just trying to connect with patients. What you need to do is identify patients and caregivers, professionals, and organizations who are aligned with you. Now, on the bottom, you can see this picture. This was our very first conference. We paid and brought together about 40 specialists from all over the US to sit in a room and really talk about some of the issues that we were facing and what we could do moving forward. That was the beginning. So first off, how do you identify patients and caregivers? If a patient comes into you and they're asking you a lot of questions, they're interested in understanding the deeper recesses of this disease, they're inquisitive about the research, they're asking you how they can help, how they or their family can help. They're interested in connecting with others, and they're forward thinking. They're not just thinking about what you're telling them right now. They're thinking about, okay, and then what will happen next and next? What's plan B, plan C, plan D? Those are the patients and caregivers who will be good advocates for you to work with. You also need to identify professionals and organizations who are aligned with your thoughts, who share the same passion that you have, and who have some time and are willing to put in some effort, because it will take time and effort. So you can see when we started, we put together a scientific and medical advisory board. We put together a nursing advisory board. That came a little bit later. And even a little bit later, then we put together a research network. And they all do different things, but they're all aligned. They all share the passion, and they all have put a lot of time and effort into building the foundations to help these patients. So one of the next things you need to do is you need to get together, like I showed in that picture of our first conference. You've got to identify what are the needs. What do these patients need? What are the gaps? What resources do you need? And then once you identify those, you can come together as a group and decide how you're going to start working on some of these problems together. You will be surprised at the ingenuity and innovation of these patients and caregivers as they work with you to solve these problems. Include them in this. They will help you solve these problems. So some possible gaps that you can think about that we thought about at the beginning is, how can we help the patients get to specialists, to providers who know how to treat this disease? Patient education materials. They need to understand the disease better. They need some information to help them navigate this diagnosis. A clinical trial finder or a clinical trial navigator, someone who can help them get access to clinical trials. Connecting patients to each other, right? Support groups, Cholangio Connect, which is a mentorship program. What would that look like for you in India if you set up a gallbladder advocacy group? And convening. Do you need to come together? Patients can help you do this. They can help you identify the way to do this. So, and then you have to identify how you're going to communicate. Are you going to build a website? Are you going to use social media? Are you going to do webinars and support groups? You have an email contact list that you'll be working from, WhatsApp. Those are some of the things that you want to do. And then also you want to identify how you're going to raise funds. And this is another place where the patients and caregivers can help you. They can help find donors. They can help you find grants. You may get funding from industry partners who want to fund something. Other nonprofits you may have other nonprofits that I or you may not be personally aware with. So do a search on the internet and see who's supporting gallbladder cancer. There may be government funding. So all of these come together just for me to share with you that there is absolutely an amazing opportunity for your group in India to work with other interested parties to create a gallbladder advocacy group. There is not a gallbladder advocacy group in the US. And we have been wondering why that no one has picked that up and run with it, but we just don't see gallbladder cancer the way that you do in India. I think it's a great opportunity for you to set something up, and then you would probably get patients and caregivers and donors from the US who would also help you, who have loved ones that are suffering from this cancer. So I hope this was helpful. Please feel free to reach out to me. We are happy to help you or provide you with resources that we have created that you could leverage and change and translate and use as you go about setting up this advocacy group. And please feel free to call me or send me an email. I'm happy to discuss this further at any time. Thank you again for inviting me, and good luck. And just know that we support you and will support your efforts in setting up a gallbladder cancer advocacy group. Thank you.
So I hope that it was helpful. I think we need to think about it, and in the next couple of months, I propose to float an email message to all of you and everyone else who's interested. Again, the most important issue will be the funding. So I would request each and every one of you to go back to your patient list and think of a patient who was related, say, to a politician, to an industry person, to a prominent person, and then we can approach them. This is how probably they also started, that would they like to start the process of helping these patients. So that's why I thought that we would include this presentation. Please.
I would now like to invite our next speaker for today, Dr. Jill Koshiol. She is also a member of the Scientific and Medical Advisory Board of Cholangiocarcinoma Foundation. Dr. Koshiol received her PhD in epidemiology from the University of North Carolina in 2005. Currently, she is the senior investigator in the infectious and immunoepidemiology branch at the National Cancer Institute at Maryland, USA. Dr. Koshiol specializes in the studies of natural history of hepatobiliary cancer and the role of inflammation in these cancers to better understand how they develop and to identify new opportunities for cancer prevention and early detection. It is my absolute pleasure to have you on board with us today, Dr. Koshiol. I would now like to invite you to present on the topic, can liquid biopsy have a role in deciding the management of gallbladder cancer?
Thank you so much for the kind introduction, and especially Dr. Kapoor, just for the opportunity to be here today. As a cancer epidemiologist at the National Cancer Institute in the US, where the rates of gallbladder cancer are very low, about 1 per 100,000 in women and half of that in men, it is a real pleasure to be here in a room full of people who are fully engaged in trying to make progress on gallbladder cancer. I'm honored to be here today. And so as we discussed, today I'm tasked with working through this topic of to what extent liquid biopsy might help aid in the management of gallbladder cancer. I thought it might be useful to begin with a little bit of a general discussion of what liquid biopsy is. I'd like to thank my colleague, Dr. Lynn Sobara from the Division of Cancer Prevention at NCI, who provided many of these general information slides. So liquid biopsy is a test performed on blood samples or other biofluids to characterize circulating tumor cells or tumor-related cells like endothelial cells or macrophage-like cells, nucleic acids like DNA or microRNAs, proteins or extracellular vessels. And people are interested in these methods because they're non-invasive or at least minimally invasive ways of trying to detect cancer, and that's always advantageous because they allow us to try to do this in a longitudinal fashion, and because we can apply it to many different biofluids at less risk to the patients. They also allow us potentially to characterize the molecular signatures of tumors that could help us better understand the biology of those tumors. So where are we in this process of applying liquid biopsy? Well, for the most part in the cancer field, liquid biopsy is used to determine recurrence and therapeutic response. Application to early detection, screening, and risk assessment typically lags behind. There is some progress being made in this area, but it is much farther behind. The typical methods that are applied include genome-wide association analysis, chip-based microarray, the SAFE-seq technology, which is now the SAFER-seq technology, and whole exome sequencing. There are quite a number of different technologies that are being developed. Some of these are currently routinely used in clinical diagnosis and decision-making in conjunction with imaging and other diagnostic modalities. One example is the FDA-approved liquid biopsy test in blood for lung cancer targeting EGFR mutations. And actually, just earlier this year, as an example of something that actually is going after screening for lung cancer, the Delphi released a screening test for lung cancer using cell-free DNA fragmentomics. So there is some progress being made in this area. There's also insurance reimbursement approved Exodex test in urine for risk assessment of prostate cancer. Potential applications of ctDNA in clinical oncology include cancer screening, and this is an area that I think all of us are especially interested in, right? How can we use liquid biopsy for early detection and early intervention? But it can also be useful for localized cancers in terms of identifying specific genomic alterations to guide therapeutic selection, monitoring tumor burden and therapeutic responses, metastatic cancer for early detection of relapse and treatment resistance, guidance of treatment selection, monitoring, etc., and even for refractory tumors to try to understand the mechanism of that resistance and potentially to determine new treatments. But despite all of this promise, there are also quite a number of challenges involved with liquid biopsy. From a biologic perspective, the target analytes are typically present in very, very low concentrations. For example, the low levels of the mutant circulating tumor DNA is diluted with circulating wild-type DNA, which means that typically you need large volumes of blood or other biofluids, and that can be a big challenge. And that's even more the case for early-stage tumors, which are shedding even lower levels of tumor DNA than later-stage tumors. There are also questions about tumor heterogeneity. So is liquid biopsy representative of all the genetic clones of a tumor? Particular clones may outcompete other clones and may be more present in the blood and therefore may not be fully representative of what's actually in the tumor. There's another issue of what you detect and whether or not that's clinically relevant. I think for gallbladder, this is not an issue. If we were able to detect gallbladder cancer through liquid biopsy, we would care and we would want to target that gallbladder cancer. But this is a concern for something like prostate cancer, for example. If you detect prostate cancer, is that clinically relevant or not? It's something to consider. And also, what is the appropriate or clinically relevant fluid? For gallbladder cancer, again, from a screening early detection perspective, I think we eventually want to target blood. But right now, I don't think we have a good sense of what those targets are. So we may want to consider looking at bile for now, because bile has been shown to have higher levels of tumor DNA. So maybe that's a better place to look right now to get a better sense of what we should be looking at. Technological challenges, again, focus on the issue of we have low levels of the genetic targets. That means there's low capture efficiency. So ctDNA detection has varied from about 50 to 78% in patients with localized disease, and there are issues with specificity as well. Other considerations include what specimens you look at, what reagents you use, what are the standard operating protocols that are applied. Many people use convenient samples, and one of the themes that has kind of come up a number of times throughout this conference is that who you look at affects the results you get. So for example, if you are looking at a bunch of different survival curves and you see separation of curves and your poor survival curves is all consists of T3 patients, of course they're going to have poor survival. So you have to consider who ends up in a study and how they got there. Who are they representative of? All of those factors have an impact on your results. Pre-analytical variables like how the samples were collected, how they were processed, how they were stored, all of those things can also affect your results. And there's also a general lack of knowledge in how to translate laboratory studies into clinical tests. CAP and CLIA regulations and certifications have a very high standard. And even if you can cross that bar, is the platform or assay cost effective? And is it portable? All of these issues are things that we have to consider to understand whether or not, in the end, liquid biopsy is something that could be useful for clinical management of gallbladder cancer cases. Additional considerations are also related to statistics and protocols. Many different studies have used different statistical approaches that can give you different answers. In the literature, there is selective reporting, typically unintentional, but that means that it's challenging sometimes to interpret what we see. Just in preparing for this talk, I saw very incomplete reporting of the protocols. For example, people often didn't report the volume of the sample that they used, and we know that the volume is important for whether you can detect something or not. It's just something basic to report, and it's not there in the published literature. There are variations in interpretation, bias, chance, overfitting. The majority of the studies published did not take their data and replicate it somewhere else. So all of these things are challenges with what currently exists, and we have to get past it to know if liquid biopsy is actually going to be effective clinically. But despite these challenges, liquid biopsy is of huge interest and growing interest. There are a number of technologies that are being developed, and here I'm showing you a whole slew of assays that are out there for using liquid biopsy to try to detect a multitude of cancers. As you can see, lung cancer is one of the most popular cancers to go after, but also liver cancer, which could include intrahepatic cholangiocarcinoma. What do we not see on this list? Gallbladder cancer. Gallbladder cancer is not here. And perhaps we would like to see it here. What has been done for gallbladder cancer? Well, here you can see a number of studies that have looked at least at biliary tract cancers. So I've listed out the number of biliary tract cancer cases for each of these studies. And then I dug through each of these studies to see, okay, well, how many of these biliary tract cancer cases are actually gallbladder cancer cases? And you can see that listed out next to it. You see sometimes it's as little as one gallbladder cancer case. This is what we have in the literature for liquid biopsy. The majority of these studies have focused on DNA targets. We do have a handful of studies that have looked specifically at gallbladder cancer, but even these studies are very small, less than 100 cases, typically less than 50 cases. And just to go into a little bit more detail on a few of the larger studies, so we have Modi et al. That included 124 patients with biliary tract cancer. This study used the Guardant 360 platform for about 70 to 73 genes and found that 76% of these patients had at least one gene alteration. But this study only included 16 gallbladder cancer cases. And the only finding that it noted for these cases is that gallbladder cancer cases tended to be more likely to have CDK6, APC, and SMAD4 mutations. That's all we get from that particular study. The next study, Chen et al., included a few more patients, 154 patients with advanced biliary tract cancer cases, looking at a panel of 150 genes. It found 95% had at least one alteration. It included a slightly larger proportion of gallbladder cancer patients. It had a kind of an interesting finding. The highest mean maximum somatic allele frequency was actually among gallbladder cancer patients as opposed to other biliary tract cancer patients. And the mutations TP53, CDK into A and EGFR were more common in gallbladder cancer patients as opposed to other biliary tract cancer patients. And then we have Burchak et al., which is the largest of the liquid biopsy studies for biliary tract cancers, with over 1,600 patients, where they found that 84% had alterations, and that those alterations were targetable for 44%. They used this to say, yay, liquid biopsy is great. We've got actionable mutations. We can use liquid biopsy for clinical management of biliary tract cancer cases. This is wonderful. But only 9% of those cases were gallbladder cancer cases. And as best as I can tell, they did not report any results specifically from those gallbladder cancer patients. And as we know, gallbladder cancer tumors are less likely to have actionable mutations than intrahepatic cholangiocarcinoma. So I actually don't know that any of these patients with gallbladder cancer had any benefit from the liquid biopsy assay that was conducted in this study. They didn't report it, I don't know. So there are a number of clinical trials that are going on for liquid biopsy for biliary tract cancer. And some of these trials do specifically include gallbladder cancer or focus on gallbladder cancer. The outcomes that are focused on in these trials include either identifying or looking at changes in circulating tumor DNA or circulating tumor cells, or at targetable mutations. But again, we know that gallbladder cancer has fewer targetable mutations than we see for other types of biliary tract cancers. So we need to see if these trials really pan out, and specifically if they pan out for gallbladder cancer. Now, I wanted to mention just a couple of other studies that have recently been published on novel targets for liquid biopsy. This paper looked at mitochondrial-free DNA in a number of different tumor types and found that cholangiocarcinomas had higher levels of circulating mitochondrial cell-free DNA compared to healthy controls. I thought that was interesting because they found that that wasn't necessarily true for all of the different tumor types that they looked at. Although keep in mind that here the numbers get pretty small. So for example, they found no difference for lung, but they only had 4 lung cancer cases. So kind of take that with a little bit of a grain of salt. I do think it would be really interesting to see what does this look like for gallbladder cancer. Do we see as strong of a difference for gallbladder as they see here for cholangio? And then another study recently published looked at serum extracellular vesicles and found that protein biomarkers in these extracellular vesicles could actually predict the development of cholangiocarcinoma among patients with primary sclerosing cholangitis, and that it could even predict early tumors, and it could also distinguish between intrahepatic cholangiocarcinoma and hepatocellular carcinoma. So I think this is really interesting. Oh, one other note about this that I thought was valuable is that the volume required is pretty minimal. Just 1 mil required in the discovery set and less than 7 microliters required in the validation set. So this really brings it into the realm of possibility in many ways. So that I think is very valuable and makes it something that I think would be super cool to look at for gallbladder cancer. So just to kind of summarize, liquid biopsy is a growing field. There is a great deal of interest, but there are a number of limitations, and we're really going to have to work together collaboratively to address these limitations. For example, NCI has a collaborative working group that can test different methods to see kind of what might be the best option, but you need to come to them with, say, 200 gallbladder cancer cases. I think you could do it here. You have that kind of capacity, but we would need to know what we want to look at. And so far, for gallbladder cancer, the sample sizes have been very small, and I don't think we have a clear idea of what targets we would want to look at. So if liquid biopsy is going to work, we need to know what we're looking for. So I think there's a lot of promise, but we need to kind of keep things in perspective with liquid biopsy as well. So with that, I will thank you for your attention, and I'd be happy to take any questions.
Thank you so much for such a fascinating talk on an emerging topic. There's very little in literature, but I think it opens completely new doors for diagnosing gallbladder cancer, especially early gallbladder cancer, and also help us manage these difficult patients. I'm sure that once we join hands, we can, bring in cases, and you can bring in technology, and together we can work towards so-called eradicating this disease. Yes, Professor Kapoor, please.
many, or I can say most of the faculty in this JSF have been my students. And for you, I have answered hundreds and thousands of questions. All of you have the responsibility to answer only one question for me. A young lady with asymptomatic gallstones comes and asks you whether I should have my gallbladder out because I am at a high risk to have gallbladder cancer. Provide me that answer. Because I don't agree that all women with asymptomatic stones should be operated, but amongst that group, the 5% population, we have to identify those who are at the highest risk. And the answer will come only from a screening test. So my request to all the young faculty members in this group is, one, create a biobank. As she said, if you have 200 samples, anybody would be willing and happy to collaborate with you. Have an idea, and then even without asking Jill, I can assure you that she will try to get a grant. I know that RO1 grants are given only for US scientists, but I know they are given to outside US also. So if you have samples, if you have an idea, it is not impossible. What I could not do, I don't want that you should also not do. One of you should take it up and do it. So first is to create a biobank and a database. Yeah, people like Safalta or the clinical. So I have a question, Jill. Now, we also have done some work on microRNA. Ratnakar is here. So with a group in Germany, they are an expert group on bioinformatics. They have done a lot of studies with Chilean colleagues also. But my question is, microRNAs have been on the scene for several years.
Yes.
But till today, I don't think for any cancer, microRNA has become a standard test. So are we following the wrong path? Should we shift our path to some other biomarker for liquid biopsy?
Yeah, I think for liquid biopsy, one of the things that people are thinking about is kind of doing a combination approach, actually. So not just DNA or just proteins or just microRNA, but trying to take the best from each of these. For example, there's a CancerSeq platform that I think Hopkins has that they're working on that includes a fairly small number of gene mutation targets along with a small number of protein targets. And my thinking is that probably something like that is also what we want to think about for gallbladder, that we don't need to limit ourselves to just DNA, just protein, just microRNAs, but what are the best, the key markers from any of those things, and how do we put them together to give us the best opportunity? Because I agree with you. I personally don't think we want to take out everybody's gallbladder who has asymptomatic gallstones, but there are some, and we need to find ways to identify the ones who are really at high risk.
But it is very encouraging to see that one liquid biopsy test has now been approved for screening for lung cancer.
Yes.
It's very encouraging.
Exactly. We're making progress.
It's just yeah, I have a thought here.
The best.
Yes. Yes. I will be taking blood as well as the tissue, and the bile will also be stored for these patients.
Yes. So
Yeah, I
here
think
I
that's
have
great.
a thought here, just one thought which comes as a gastroenterologist. So when we are doing ERCP, that means when we are going into the bile duct and taking bile from there in the process of putting in stents. So that time we can access bile, because by the time the circulating tumor DNA is coming to the blood, it's going to take a long time. And by that time maybe the disease has already started spreading. So the best time to look at it is to look at the bile in the duodenum, where you give a CCK infusion and then the bile comes into the duodenum. You can take that and look for the tumor DNAs, or during ERCP in patients who are undergoing a procedure for gallstones. So there we can track gallstones which are in a 20-year-old person and a 50-year-old and a 70-year-old, and to see whether the DNA start increasing as age goes up, and what are the parameters which decide that. So, and also Dr. Pankaj is here, so we do PTBD, that is, we put in a catheter into the bile duct in patients with obstruction. So that's another place where we can get bile and look at it directly, and then identify which are the RNAs we are looking for, which are the DNAs we are looking for, and then look for them in the blood.
Yes,
So that may be
yes.
good.
Do you think that the looking in bile from the bile duct itself versus bile from the gallbladder will give you different results?
It may, but once you, in patients with CA gallbladders, we put in a catheter into the bile duct. So then it forms a path of least resistance. So the bile is going to now start flowing through that. So I'm sure we'll be able to access sufficient DNA tissue in that, and maybe we can plan out such a national project on this aspect.
Fantastic.
So do we have any other quick questions or comments from the house? If not, I'll thank you, Jill, for a wonderful lecture, and thanks for the fascinating journey to look forward to. Thank you.
Thank you all.